Hello, welcome student. We shall start a new point, that is LAC-OPERON. Understand? It is this system was found in the prokaryotes. Found in the prokaryotes, and that was first described by Jacob and monads that was first described by Jacob and monads in 1961. That is called and found in the prokaryotes. Why it is found in the prokaryotes? We have just studied a day before that in the DNA or RNA segment which are found, it contains multiple genes. Means the system is controlled by multiple genes. That is polycystonic. So, and while in eukaryotes, it is monocystonic. So this is found in the prokaryotes due to polycystonic nature. Due to polycystonic nature, it is found in the prokaryotes. Understand? And now, what do you mean by LAC-OPERONs? LAC means lactose. Why it is called lactose? LAC-OPERON means lactose operation. And lactose, you know, it is a disaccharide. Lactose is a disaccharide, means it is composed of two monosaccharide units. One is glucose plus galactose. It is composed of two monosaccharide units, that is the carbohydrates, and that is and glucose, you know, that is the source of energy. So this system LAC-OPERON, it is a, just I have studied told you in the previous video that it is a inducible operon. Means normally it is in off conditions, but and then on necessity it starts, that is, it goes in on position. And this off and on positions, it is regulated by lactose. It is regulated by lactose. And so lactose is the inducer of the system. So lactose is the inducer of the system, is controlling on the necessity, it on the system, and when there is no necessity is off the. Suppose that it is a prokaryotes. Now here is a medium that contain glucose, If the glucose is present in the medium, and galactose is also, so the prokaryotes will want to take glucose for its energy. That in that position there is no necessity to take the lactose. Means in presence of lactose and glucose, the glucose will be preferred by the prokaryotes. अगर लैक्टोज और ये ग्लूकोज दोनों ही मीडियम में हो समझे, तो वो जो है प्रोकैरियोट्स ग्लूकोज को लेना पसंद करेगा समझे। और जब ये ग्लूकोज लेगा लैक्टोज नहीं लेगा तो उसमें ये सिस्टम का होगा, दैट विल बी इन ऑफ कंडीशन। मीन्स इट्स नॉर्मली इन द ऑफ कंडीशंस। मीन्स क्योंकि ग्लूकोज से ही उसको एनर्जी प्राप्त करना है समझे। he will take the energy from the glucose, and if the glucose is available in the medium, so it will prefer to take the glucose instead of lactose. Now we see how the system goes on. First we will see when there is no lactose, means in absence of lactose, when the medium has sufficient glucose, in that conditions the system will not go ahead, and so it is in off conditions. Means when lactose is absence, that is in the condition of off. The system contains this is the P, this is PI, this is P, this is O, this is Z, this is Y, this is A, capital and small, whatever you can write. These are the some stages of the system LAC-OPERON. P, promoter gene, I, inhibitor protein, O, operator gene, Z is beta galactosidase, Y permeate, A trans-acetylase. These are the following steps of the operon. That is promoter, inhibitor, that is promoter, that is operator, and these are the ZYA, they are the structural genes. So now on the P means promoter, the mRNA will entered. This is also will enter. This is the operator, understand? And this is Z. It will control whether the mRNA will be allowed to go in this side. Now this. So promoter, inhibitor, this is promoter, this is operator, this. Now promoter, inhibitor, operators in galactosylase. One thing, it is galactosylase. Responsible for the hydrolysis of disaccharide, that is of lactose, because we know that lactose and this permease enzymes increase the permeability of cells, and so this is the inhibitors, inhibit the protein synthesis. This is the promoter and this is the operator. Now we see how the now we know that in the central dogma we have studied that mRNA by means of translation forms the protein. You already know, just keep it in mind. Now it is the just when mRNA will go towards that side, means will cross the operator gene. Means operator gene, if the operator gene allowed to mRNA to go to the structural genes, that will go that and the transcription will be take place. And if it will not allow, then the translation will not take place. So this first inhibitory genes, it pressure and finally it forms a protein, and with this impressor is binds the operator gene. It binds the operator gene. And this binds the O gene. So mRNA will not allowed to go forward for translation, will not allowed to go forward for the translation, and no protein will be synthesized. So this is in the case of absence of in the off position of lactose. Now if the lactose is present, lactose is present, that is in the on condition. This protein which is formed, it entered the system through permeases. Enters the system through permeases, and at the same time the lactose will enter and the protein plus lactose will combine to each other, and when the lactose will go, it will not bind with operator. Then it will not bind with operator, means, and so the mRNA will go in that system and in that system, and now due to that the lactose changing glucose and galactose. Glucose and galactose, understand? And that glucose will be used for the prokaryotes, and in the way the lactose will be entered one by one till it is ended. When the lactose will completely ended, the system will not go ahead and it will off. So it said that, yeah, it clears that when the lactose entered, it will not allow to increase the messenger RNA, so it will allow to the messenger RNA to go towards the structural genes, and the process is on. इससे पता चलता है कि जब लैक्टोज इसमें जाता है तो इनहिबिटरी प्रोटीन्स को ऑपरेटर के साथ मिलने नहीं देता है, जिससे क्या होता है मैसेंजर राइबोन्यूक्लियस विल गो अहेड एंड डू इट्स वर्क. और जब लैक्टोज नहीं होता है तो यहाँ जो प्रोटीन्स बनता है दैट विल इनहिबिटरी प्रोटीन्स होता है, जो ऑपरेटर्स को जाकर क्लोज कर देता है, समझे, ताकि मैसेंजर राइबोन्यूक्लियस विल नॉट गो अहेड एंड द प्रोसेस विल नॉट कंटिन्यू. सो दिस सिस्टम इज कॉल्ड LAC-OPERON.